Okay, let's go ahead and get started. It happens every year. I've got my first few emails about what do we study. And if it were me, assuming I'd been foolish enough to let the drop add date slide, what I would do, so you've got the study guide, and if you haven't been going to the lectures and going through that material, I don't think it'll make any sense. And the idea of the study guide was to reorganize the material a little bit. And if it were me, what I would do is I'd take the names of all the hormones and write them down on a 3x5 card, individual 3x5 cards, shuffle them and sort them as many ways as you can think of. You know, obviously, which are the proteins, which are the steroids, you know, which ones of the proteins, which ones bind to G-alpha I's, which one binds to receptor tyrosine kinases, things like that. So with that, let's go ahead and finish up cortisol and the slow stress response. And the last thing I went over was the mechanism for signaling for glucocorticoids. Okay. And a lot of what we know about mammalian transcriptional regulation was actually worked out on glucocorticoid receptors before anything else. So quite a bit is known about them. Okay. So what's important to remember, the take-home lesson is transcriptional viability is determined by histone acetylation state. That the glucocorticoid receptor cortisol complex associates with histone acetyltransferases or histone deacetyl transferases and other transcription factors to modulate transcription. Deacetylases, not deacetyltransferases, sorry. And the result is large-scale transcriptional modulation. So that, as a general rule, even though sometimes we present it that way, steroids don't activate transcription of a couple of genes. They often activate transcription or repress transcription of hundreds, or in the case of cortisol, maybe even thousands of genes. So the question becomes, is this a general mechanism for all steroids? And again, an awful lot is known about the way cortisol works. But I think this is probably a general mechanism that will eventually apply to pretty much that entire family of nuclear receptors, nuclear steroid receptors. Okay, so you should be at least a little stressed now. Okay, and we haven't even talked about what cortisol does. Okay. So cortisol mediates the slow stress response. Okay. So it has metabolic effects, and these are mostly catabolic. Okay. The one exception is that it promotes glycogenesis. Okay. Cardiovascular system, it stimulates cardiovascular output and red blood cell production. Forget about this thing with calcium. The source I originally consulted had the story messed up. Let's just leave that aside. It's not quite what your book says. Okay. In the kidney, it helps to conserve water. Okay. In the anterior pituitary, it inhibits luteinizing hormone and FSH, follicle stimulating hormone, FSH. Those are both involved in reproduction. So as you might imagine, it inhibits reproductive functions. It may inhibit growth hormone, although that's a little questionable. And it inhibits thyroid stimulating hormone. It also has really important psychological effects. I think next year I'll probably go into this a little more. But especially long-term, cortisol has, and especially in children, cortisol can have long-term elevated levels of cortisol, can have really profound psychological effects. Now, I talked about a slow response. There will be a fast response, and that'll be mediated by epinephrine, which we'll get through in the next topic. Okay, so the best known response for cortisol, or glucocorticoids in general, is homeostasis of inflammatory immune response. And this will let you know why we stick with the terminology of glucocorticoids. Okay. So the immune system, of course, responds to injuries or various kinds of stresses. Okay. And the first line of defense in the immune system is inflammation. Okay. It involves synthesis of prostaglandins, a local defense response that involves clotting and swelling and pain, okay, that helps to inhibit infection or bleeding. Okay. The problem is that certain kinds of stresses can promote a runaway immune response. So that in small doses, these things are great. In large doses, they can be a real problem for homeostasis, getting a response way out of proportion to the actual insult. Okay. Cortisol inhibits the stress response to reestablish homeostasis. So people get this confused. They think, aha, cortisol and stress response, and think it ought to promote immune function. It doesn't do that. The immune system doesn't need the endocrine system to work. What it needs the endocrine system for is to back down. Okay. And remember, cortisol is slow. Inflammation is very fast. Okay. So the inflammation will happen, and then the cortisol will come in afterwards and inhibit that initial inflammatory response. Okay. So the first thing I mentioned was, of course, metabolic. And cortisol ensures glucose for the central nervous system. So here we have our liver and sort of a beta oxidation setup. Now, when we've talked about metabolism, nutritional state has been key. But when it comes to cortisol, that's not important. Okay. So the mechanism is it increases enzyme synthesis. Since it works on transcription, okay, all it can do is increase the synthesis of certain metabolic enzymes, which of course is slow. And the key is in liver, where it increases transcription of enzymes involved in gluconeogenesis to allow more production of glucose 6-phosphate, glucose, and maintain blood levels. Okay. It also increases glycogenesis, okay, to store glucose for later. It often opposes the activity of insulin. Okay, so where insulin is giving you an anabolic response, most of what cortisol does is catabolic, okay, except in glycogenesis. That's the one key difference, and that's one of those nasty things you're probably going to have to memorize. Okay, so it works together with insulin. All right. Other metabolic effects support liver glucose synthesis. Okay. So, for example, it stimulates protein catabolism, increasing available amino acids for gluconeogenesis. It inhibits the synthesis of GLUT4. Okay, so it doesn't interfere with the way it's transported to the cell surface, but it does inhibit its synthesis, so less glucose can be taken up in the muscle and the adipose. Okay. It increases hormone-sensitive lipase to promote the conversion of non-esterified fatty acids for energy for gluconeogenesis. So again, it doesn't directly activate it, it just increases the amount, to increase the amount of non-esterified fatty acids. And it also inhibits the transcription of insulin, as well as genes required for insulin secretion. Cortisol is regulated through the hypothalamic pituitary axis. From the hypothalamus is corticotropin-releasing hormone, or CRH. Okay. Again, this is one of these things, it's not hard to memorize because it has cortisol in the name. So if you kind of got the base hormone, this will give you, the corticotropin should lead you to cortisol, releasing hormone should lead you to the hypothalamus. Okay. It's a 41 amino acid peptide. It's stimulated by stress sensors in the central nervous system. It's G-alpha S coupled. It stimulates the release of adrenocorticotropin. We usually call it ACTH because adrenocorticotropin is a mouthful. It's, of course, from the anterior pituitary. It's a 39 amino acid peptide. We ran into it before. It's one of the products of pro-opiomelanocortin. And it's also G-alpha S coupled. So CRH and ACTH increase cortisol synthesis. All right, so CRH here binds to the G-alpha S receptor, activating adenyl cyclase, and giving us active protein kinase protein kinase A. Okay. That directly causes the opening through phosphorylation of a calcium channel, and then that calcium promotes release of ACTH. ACTH also binds to G-alpha S receptor in the zona fasciculata in the adrenal cortex, giving active pKa. And the pKa does a lot of things. It directly activates or directly stimulates the activity of steroid acute regulatory protein and cytochrome P450 side chain cleavage. It also activates a transcriptional program that increases synthesis of steroid acute regulatory protein, increases synthesis of the side chain cleavage enzyme, as well as the other steroidogenic P450s, it also increases synthesis of the LDL receptor, okay, and critically, the size and number of adrenal cortex cells. Okay, so the adrenal cortex can't be maintained in the absence of ACTH. If you have it, you have a big adrenal cortex. If you have a lot of it, you have a much bigger one. If for some reason ACTH gets cut off, the adrenal cortex will atrophy. Okay, now this is sort of the classic for negative feedback loop. So we've got CRH, ACTH, and this is cholesterol in the adrenal gland, in the adrenal cortex. So emotional stresses, time, there's an important circadian component to CRH release, and things like infection, trauma, hypoglycemia all activate CRH, all activate the release of CRH, which activates the release of ACTH. Thirst is another important stress, but that works through antidiuretic hormone, which we'll also get to today. ACTH then stimulates both directly and through transcription the synthesis of cortisol, which is released from the adrenal gland. Cortisol feeds back negatively to reduce release of ACTH and CRH. ACTH also feeds back negatively to CRH. This is really the classic short and long loops negative feedback system. And CRH is also inhibited by homeostasis. So good feelings, happy thoughts, being comfortable and happy and fed and all that inhibits CRH. Okay. So circadian rhythms are an important regulator of cortisol. So circadian rhythms are our natural biological day-night cycles. And so on this graph, we've got cortisol here, and that's in blue, the blood levels. And over here, we've got ACTH in red. You see ACTH is pulsatile, right? And the cortisol peaks early in the morning, so after a series of ACTH pulses, which are higher amplitude, and then tends to drift off down toward midday. The ACTH pulses continue. They will drop dramatically in the evening, okay? And that dramatic drop in ACTH pulses causes a drop in cortisol levels at night. So your cortisol levels should be low at night. Now, I used to put more emphasis on the whole circadian rhythm thing. And one of the things that's made this story complicated is we now know that the liver actually has its own circadian rhythm. So it's actually built into the transcriptional program of cortisol, as well as being regulated through the hypothalamic pituitary axis. Okay, so note that the hormone levels are higher in the morning, and that the reason, I said the basis, that's not right. The reason is not understood. Okay, what biological advantage there is to having high cortisol in the morning. Okay, and that the secretion of ACTH is pulsatile, like the other hypothalamic and pituitary hormones. Okay. Also note that there's a lag here between the response of cortisol and the pulses of ACTH. So that even when ACTH is very low, cortisol is very high here. Okay, and that's because of the transcriptional regulation required to make cortisol. Okay. So corticosteroids is drugs. Okay, so we call them corticosteroids or glucocorticoids. Okay, and they're probably best known for their pharmacological effects. Okay. They're a very effective anti-inflammatory. Okay. So they can control the swelling and the pain caused by runaway inflammation. Okay. They also control allergies and autoimmune diseases, things like arthritis, rheumatoid arthritis, inflammatory bowel disease, lymphoma, leukemia, probably most critically asthma. Okay. And it turns out at any one time, 0.5% of the entire United States population is on glucocorticoid therapy. Okay. So that's 1.5 million people, actually more than that, about 1.6 million people. So it's probably worth thinking about a little bit. Okay, so there's some complications with corticosteroid therapy that teach us a little bit about endocrinology. So here's our HPA system with the feedbacks put in. Okay. This is one example of a corticosteroid. There's a lot of them. Okay. And those of you who want to go to pharmacy school will get these much more extensively than I have time to present. Okay. And the effect of high-dose corticosteroid therapy, as you might expect, is to get into this feedback system and inhibit synthesis of ACTH and CRH. So this whole CRH-ACT cortisol axis is suppressed. Okay. With the suppression of synthesis of these two hormones, the synthesis of cortisol, native cortisol, is also suppressed. Over the long term, that will cause adrenal atrophy. There are also suggestions that it will actually, because we saw how radically cortisol could change chromatin structure, that there's remodeling in the pituitary and in the hypothalamus, which makes it very difficult to just restart, for transcription to restart synthesis of CRH and ACTH. It can take weeks to recover from this. And so slow withdrawal of corticosteroid therapy is always necessary. So if a person's on it for several weeks, they need to carefully ramp down their dosages over time to allow the whole HPA axis and the adrenal gland to recover. Otherwise, they won't have any normal cortisol response and will really be leaving themselves open to a variety of runaway immune responses. So the pathophysiology is pretty well known. There's hypercortisolism, goes under the name Cushing's disease or Cushing's syndrome. Okay. Now, it's a syndrome is a collection of symptoms. Okay. And it's usually iatrogenic, which is a fancy word for saying the doctor did it, which isn't really that fair, from corticosteroid overdose. And so what happens is that people end up taking too much of these, or too much is prescribed. Okay. If it's Cushing's disease, if it's considered Cushing's disease, it's due to usually a primary disease of the adrenal gland, okay, caused by a tumor or other influences from the adrenal or pituitary gland. Okay, and Cushing's disease is pretty rare. It's much more often the syndrome. Okay. A secondary Cushing's disease, the secondary causes might be due to a pituitary tumor. So a tumor that oversecretes ACTH. There's also hypocortisolism, which goes under the names Addison's disease or syndrome. Okay. And this Addison's disease is usually also iatrogenic, that if an individual withdraws from corticosteroids too fast, their own response won't pick up right away, and that'll cause Addison's disease or Addison's syndrome. The disease is rare. Okay. And it's due to an autoimmune attack on the adrenal gland. Okay. And we'll go over the symptoms and the effects of the hypercortisolism, but hypocortisolism is kind of complicated and really doesn't teach us that much about endocrinology. So you can either read about that on your own or get into medical school and they'll tell you all about it. So Cushing's disease, okay, or syndrome. So cortisol will cause transiently, so transient stresses that result in cortisol release will depress appetite. Okay. But chronic stress leading to chronically high cortisol levels increase appetite, and overall cortisol levels end up being reset at sort of a higher base level. Okay. Cushing's is the maladaptation to chronic stress and increased cortisol levels. Okay. It's a diabetes-like condition, and you'll see why in just a second. It's characterized by a lot of complicated symptoms, but most prominently an increase in adipose mass, especially viscerally. It's also muscle wasting. And there's other symptoms. So here's a picture of a person with Cushing's disease or syndrome. They have this she's got this characteristic moon face, coloration in the cheeks, okay? And there's a real tendency to not only gain weight, but gain it abdominally. Remember we talked about that maladaptation in diabetes. And stress is one of the causes of it. Okay. So the way to think about Cushing's syndrome is that individuals end up in the fed state, but with chronically high cortisol. Okay, and it involves the interaction of mostly insulin and cortisol. Okay, so what's important to remember is that the cortisol effect can defeat the insulin effect, or at least fight it enough to really depress it. So we have insulin causing synthesis of glucose 6-phosphate, glycogenesis, glycolysis, as well as inhibiting gluconeogenesis. Now, cortisol stimulates synthesis of enzymes involved in glycogenesis. So this is actually a synergistic effect. So there's glycogen storage is enhanced. However, it also stimulates synthesis of gluconeogenic enzymes, okay? Increasing synthesis of glucose 6-phosphate, and as a result, glucose. And so glucose, you tend to have more of a gluconeogenic effect in the liver. Insulin directly stimulates the transport of GLUT4 to the surface of skeletal muscle cells. But cortisol decreases the transcription of the GLUT4. So while the insulin signal still functions, there's just not as much GLUT4 there to be transported to the surface. So glucose uptake into the skeletal muscles is inhibited. Insulin promotes an anabolism of proteins and inhibits catabolism. But the strong catabolic signal of cortisol will diminish this, causing increases of amino acids used for gluconeogenesis. In the adipose tissue, again, we've got this direct stimulation of GLUT4 by insulin, GLUT4 transport, which is dampened by reduced transcription due to cortisol. Now, of course, insulin stimulates the transport of lipoprotein lipase to the cell surface and the whole lipid anabolic pathway. Okay. Because glucose tends to be high in Cushing's syndrome, insulin levels tend to be high. So this anabolic signal is very strong. And that's what causes the increase in lipid storage, the increase in adipose mass associated with Cushing's. Now, cortisol also increases the transcription of hormone-sensitive lipase, which then needs to be directly activated by glucagon to be active. However, in the presence of high glucose and high insulin levels, glucagon isn't secreted at high levels. So even though there's more hormone-sensitive lipase to give you this catabolic effect, the lack of the glucagon signal keeps the hormone-sensitive lipase levels low, or activity low. All right? So the metabolic effects of cortisol explain Cushing's syndrome. So cortisol mediates all its effects through transcription. Some of insulin's effects are post-transcriptional. Okay? So the transcriptional regulation overwhelms that insulin signal. Insulin still works, but there's just much less of the enzymes for it to activate. The result is hyperglycemia due to increased blood glucose levels from liver gluconeogenesis, decreased GLUT4 transcription. The muscle wasting is due to cortisol pushing catabolism over anabolism. And the increase in adipose mass is due to high insulin levels, increasing lipid anabolism, combined with low glucagon levels, depressing lipid catabolism. So that's kind of just the metabolic effects of Cushing's in a nutshell. Okay? So that's the end of cortisol. And let's try to do a top hat question. This one's going to be on oxytocin. And we'll see if it works this time. There we go. Here we go. All right, so wait a second. So it's a peptide hormone from the posterior pituitary. Identify the structure from where oxytocin is secreted. There we go. So an E is none of the above. So oxytocin is an anterior, this is just not a good system. All right, let me see if I can do it this way. No, it doesn't work. So oxytocin is a peptide hormone secreted from the posterior pituitary. Identify the posterior pituitary. So it's A, B, C, D, or none of the above. Which one's the posterior pituitary? Yeah, it didn't work. Eight seconds. 5 seconds. Okay. Show answer. So did everybody get D? We'll see. Okay, good. Yeah, I think that's about the, I can, so C is, I think, C is the anterior pituitary. If you did A or B, you need to start studying a little more, really. I mean, it's getting late. There's a, no matter what, there's a lot of material in this section. I can't help that's the nature of this area. And so you need to be able to get stuff like this in your head. 91% I think is about what the class did when this showed up on the test. So. All right, good. So, and with that, we will move on. All right. So now there was a slow stress response, so there kind of had to be a fast stress response. And that's mediated by a hormone called epinephrine. It's made the adrenal medulla, and we'll go through the usual things, synthesis control, and there's a lot of diversity in responses from epinephrine, which are worth sort of walking through. So epinephrine is a catecholamine, which is a kind of amide hormone. Okay? You can see it's not anything like thyroid hormone. It's classified as an amide hormone because it's made from an amino acid. It is made in the adrenal medulla, the very inside of the adrenal gland here. The cells it's made in are called granule cells, okay, because it has these little things called granules. Okay. So technically, the adrenal medulla is not considered a true gland. Okay? And in some animals, it's separate from the rest of the adrenal gland. Okay. The reason it's not considered a true gland because it's really more like a specialized sympathetic ganglion from the nervous system. You doing okay over there? This is exciting, right? So your heart started beating real fast, then it slowed down. Yeah, that's epinephrine. Okay? So anything that scares you, you sort of get startled and it slows down. That's epinephrine. I actually used to yell at the class and chew them out for doing such a bad job and threaten a pop quiz. And that all lost its edge once we started doing these questions as a demonstration for epinephrine. Okay. Anyways, the blood supply here is important. So here's the veins and the arteries. Okay. And it goes from the cortex to the medulla. Okay. And this is another case where the anatomical structure lets you know what kind of regulation is possible. Okay. So that cortisol can regulate synthesis of epinephrine in the medulla on the inside, but the opposite couldn't be true. Okay. Epinephrine is a stress hormone. Okay. The adrenal medulla is an extension of the autonomic nervous system. So it connects the endocrine system with the sympathetic nervous system. So things like trauma, anxiety, hypovolemia, hypoglycemia, pain, hypothermia are sensed in the brain. Signals are sent down the spinal cord into a preganglionic sympathetic nerve, which releases acetylcholine, and in response, epinephrine is released from the chromaffin cells of the adrenal medulla. Okay. This is the fast endocrine response. This happens very quickly. You get startled. Immediately your heart starts beating faster and stuff. That's due to epinephrine. Epinephrine works with the sympathetic nervous system through the neurotransmitter norepinephrine. Okay. So they're activated by the same kinds of stresses. Okay. In the case of epinephrine, we have the preganglionic neuron to the medulla. In the case of norepinephrine, we also have a preganglionic sympathetic neuron, which then leads to a postganglionic sympathetic neuron. Of course, releases acetylcholine, and in response, releases the neurotransmitter norepinephrine, which is very similar. If you look at the structures, it's just this methyl group. Okay. So norepinephrine is not released into the blood. It's right on top of the tissues it works on. It's not released in the blood a lot. Okay. Its blood levels will be pretty low. Okay. But epinephrine and norepinephrine almost always affect the same tissues. Okay. And for that reason, their effects are really hard to separate in physiology. It's really hard to be sure what's due to one and what's due to the other. So medically, they're generally referred to together as catecholamines. Okay. And this represents the integration of the nervous system and the endocrine system to conduct the fast stress response. So epinephrine is made from tyrosine in chromaffin cells, okay? Just like thyroid hormone, but by a much different kind of biosynthetic pathway. So it starts with tyrosine, and this is free tyrosine now. Okay? It's not attached to a protein. It's not part of a peptide chain. Tyrosine hydroxylase converts it to dopa, amino acid decarboxylase, to the neurotransmitter dopamine. It then enters the granule through a catecholamine proton exchanger. Okay. Those protons are provided through a proton pump in the granule membrane. The dopamine then is converted by dopamine beta hydroxylase to norepinephrine, which through a process that's not understood leaves the granule cell, is converted to epinephrine by phenylethanolamine N-methyltransferase. Epinephrine then is pumped back into the granules by the same transporter. Okay. And it complexes with a protein called chromogranin, which collects the epinephrine and binds it. Okay. And that serves to concentrate the hormone inside the granule, so that the epinephrine concentration inside these granules can be up to 0.5 molar. Right? And that allows the epinephrine to concentrate and be released all at once. Okay? Don't worry about the names of these enzymes. And really don't really worry about the pathway. Okay? Just know that there's a pathway and it's something like this. I'm more interested in the regulation. Epinephrine synthesis is regulated by the CRH-ACTH cortisol axis. Okay? So we have corticotropin releasing hormone, ACTH, cholesterol to cortisol, the adrenal cortex and medulla. Okay, so we have our stresses, which stimulate the release of CRH. And we have now, this is the entire negative feedback and synthesis regulation for cortisol. Okay. Cortisol stimulates the transcription of some of the enzymes involved in the conversion of tyrosine to norepinephrine. And ACTH stimulates the biosynthesis of other enzymes in that pathway. For release, then, it's the sympathetic stimuli is what ends up causing release. Okay, so and that's through ACTH, and that stimulates release of chromogranin and epinephrine from the granules into the bloodstream. Okay. Note that epinephrine is not directly involved in any sort of HPA feedback. So it's part of the system, but it doesn't directly inhibit the synthesis of CRH or ACTH. Okay. This regulation is integrated at the cellular level. Okay, so we start with ACTH, which activates adenylyl cyclase, which gives us active protein kinase A. Protein kinase A then works through a transcriptional program, not through a program involving direct effects. And that transcriptional program increases transcription of one of the enzymes involved in tyrosine to dopa conversion, or of the enzyme, and of the conversion of dopamine to norepinephrine, the beta carboxylase. Cortisol then activates a transcriptional program that leads to biosynthesis of another enzyme. Okay? And again, I don't name these enzymes. You don't have to know the exact enzyme, just they're working at different spots. Release of epinephrine from the granules then happens through ACTH, or ACH, acetylcholine, from the sympathetic neuron. It binds to a channel called the NMDA receptor. Okay? And the NMDA receptor allows sodium to run into the chromaffin cells. Okay? That changes the membrane potential. That change in membrane potential activates a voltage-activated calcium channel. Calcium comes into the cell, and it's then calcium that causes the release of the epinephrine chromogranin complex. And as soon as it's into the bloodstream, they dissociate. Chromogranin then is destroyed by a protease called plasmin. It's attached to the cell surface here. And it appears that those fragments of plasmin can then negatively feed back and inhibit further release of epinephrine. Epinephrine has a very short half-life, okay, 1 to 2 minutes. And again, you get that startle response, and your heart starts beating real fast, and you can sort of almost feel the 1 to 2 minute half-life as it slows down again. Epinephrine is metabolized by catecholamine O-methyltransferase, primarily in the liver. Okay. All right, so it puts this methyl group onto the aromatic ring here. Okay. Also in the vascular endothelium, where a lot of the effects of epinephrine occur. Okay. And importantly, also in the heart and the kidney. Okay. In the liver, then, metanephrine is then converted to vanillomandelic acid. Okay? And then that can be glucuranated or sulfated in either the liver or the kidney and excreted. Okay? So this is a lot like the degradation of metabolism of steroids. Okay? So in that regard, in its synthesis and in its degradation, epinephrine is very much like a steroid. Its release mechanism is more like a peptide. So epinephrine and cortisol work together. Right? So we have a stress, and there's a fast stress response. Okay? And that involves sympathetic nervous system release of acetylcholine from sympathetic ganglia, epinephrine release from the adrenal medulla, and norepinephrine release from the sympathetic nervous system. There's also the slow stress response. Okay? That involves ACTH and cortisol upregulating epinephrine synthesis through transcription of synthetic enzymes, packaging into chromogranin, and feedback through the ACTH CRH system. And it's important to remember, epinephrine is not involved in that feedback. So this is more a response of that system. Okay? So you have two methods of regulation that proceed at different rates. Catecholamines bind to G protein coupled receptors. So in that regard, they're like a short half-life, more like a peptide. Bind to G protein coupled receptors, like a peptide. Okay? These are called adrenergic receptors, because when this response was originally called the adrenergic response, because it was known to occur from something coming from the adrenal gland. Okay? These receptors were originally defined pharmacologically. So there's a lot of different compounds that bind to these receptors. Okay? And as it's turned out, some of the names have actually stuck once we got to know the genomics of it. Okay? There are beta 1, 2, and 3 receptors, which are all G-alpha S coupled. There's the alpha 1, which is G-alpha Q coupled, and alpha 2, which is G-alpha I coupled. Okay. They vary in their tissue distribution. And that's the key to understanding the multiple effects of epinephrine. Epinephrine does a lot of things, and what it does is defined by which receptor is in the tissue. Okay. And there are multiple subtypes of each. I mentioned there are 3 beta receptors, which are quite a bit different from one another. There are also 3 alpha 2 receptors, A, B, and C, alpha 2A, B, and C, and alpha 1, A, B, and D. Turned out C wasn't real, even though pharmacologically it seemed that way. All right? So you're not going to have to worry so much about the difference between these. There's 9 of them, and it may come up in a question, but not in a way where you have to just generate, which one is G-alpha I coupled. Okay. Now the alpha receptors have a higher affinity for norepinephrine over epinephrine, and the beta have a higher affinity for epinephrine. People in pharmacy school love to point that out to me, but it doesn't really help you understand the effects of the hormone. It's just a sort of pharmacological thing. And even though I tell you don't worry about the identity of these things, I know some of you are trying to get into pharmacy school, and if you succeed, you will get these more than you'll be sick of them, trust me. Or fascinated by them, one of the two. I hope the latter. Okay. So the major effects of epinephrine involve what has come to be called a fight or flight response. Okay, so it's an emergency stress situation. So the alpha 1 receptor causes smooth muscle contraction, which will decrease blood flow to the organs around that vasculature. So smooth muscle contraction in the vasculature, okay, cutting off blood to the spleen, the GI, the bladder, and to reproductive organs. All places where not much blood, that are not high priority tissue systems in a fight or flight situation. If you've got to fight the bear, these things just don't matter. The alpha 2 receptors are in the central nervous system mostly and are known to decrease pain. They are also located in the pancreas, where they stimulate the release of glucagon and depress the release of insulin. The beta 1 receptors stimulate cardiac output from, of course, the heart. The beta 2 receptors promote smooth muscle relaxation to stimulate blood flow in the vasculature, okay, importantly to the heart, so cardiac blood flow. These are high priority tissues. So the heart, you might guess, skeletal muscle, increases blood flow to the skeletal muscle. It increases blood flow to the lungs. The lungs also have beta 2 receptors in their bronchia, so they can, the air capacity of the lungs also expands. And they also stimulate gluconeogenic enzymes directly in the liver, beta 2 receptors. Beta 3 receptors appear to be mostly in the adipose tissue, where they stimulate hormone-sensitive lipase, leading to lipolysis. So epinephrine mobilizes glucose. Okay, so down here we have our skeletal muscle, liver, adipose tissue, pancreas is down here in the corner. We have insulin. One of the first effects of epinephrine is to inhibit release of insulin. It also stimulates the release of glucagon. It activates hormone-sensitive lipase to increase its activity and increase non-esterified fatty acids in the blood, and inhibits GLUT4 transport to the adipose tissue surface. These amino acids can then be used for gluconeogenesis, as well as, oops, it's in the wrong order. Okay, there's amino acids for gluconeogenesis and fatty acids for beta oxidation, increasing the pyruvate, stimulating gluconeogenesis, increasing the amount of glucose 6-phosphate. Okay. It also stimulates glycogenolysis, right, increasing the amount of glucose in the bloodstream. The skeletal muscle, it inhibits GLUT4 transport to the membrane surface, stimulates glycogenolysis for ATP production. High levels of beta oxidation, in turn, will increase ketone production, to continue ATP production in the muscle because either you're fighting or running away from the bear. But most of this is, again, the part of the stress response is to make sure that the central nervous system stays supplied with glucose in the presence of all these other stresses that are sending blood and metabolic fuel to other tissues. Now, glycogen metabolism differentiates cortisol from epinephrine. People always get tripped up on this. Right? So here we have glycogen, glucose 6-phosphate, and glucose. Okay. Cortisol promotes glycogen synthesis to ensure long-term supply of glucose to the central nervous system in times of stress. Epinephrine promotes glycogen breakdown to ensure the short-term supply of glucose to the central nervous system during times of stress. Okay, so this is the one place where the effects of epinephrine and cortisol are opposing each other. Okay, so that's the fast stress response. And in the end, what you get is kind of a picture of how the endocrine system and the central nervous system work together to promote this fight or flight response. And to the extent people believe in this sort of stuff, this is really where the mind-body connection comes from. So here we have the sort of major players. So in response to an injury or an emotional stress, injury promotes inflammation. Injured tissues release cytokines, which also promote emotional stress. So any of these end up sending signals to the hypothalamus, which promotes these stress hormone releases that we've gone over. Okay. These are nervous functions. These are peripheral functions. Okay. Epinephrine and cortisol will both mobilize glucose. Norepinephrine and epinephrine stimulate a cardiovascular response. Cortisol will too on a slower scale. These will both inhibit visceral functions. This is the reproductive tissues, GI. Okay. Norepinephrine from the sympathetic nervous system also inhibits hormones involved in growth and reproduction, as does cortisol, as well as behavior leading to sexual activity. But norepinephrine stimulates arousal and aggression. In the end, there's a feedback to reduce ACTH and CRH. These are all self-correcting. Cortisol inhibits inflammation to reestablish homeostasis. Okay, and again, that's not something you need to sit down and memorize. I'm just trying to get you to sort of see how these things go together. Okay, so we have time for a couple more questions. Oh, here's my little top hat thing. Oh, here we go. Okay, so so this is supposed to be G alpha S. And this is G alpha I. I apologize for that. It looked better on my computer. So again, A is G alpha S, B is G alpha I. Next time I'll forget about their codes and just spell it out. Okay, you shouldn't be looking at your notes here. Really. Really, it's better for you if you just think this through. Okay, who got this right? Some people just are not very confident in this. All right. Okay, so it's G off I. So of those who thought you got it right, how many of you were correct and actually did get it right? Okay, good. All right, good. Yeah. So this is the kind of, you see, this just doesn't work very well. All right. I'll write them again and complain. This is the latex code you're supposed to put in to get the equations. So it's from the hypothalamus. You probably haven't gone on overgrowth hormone. I put this example in this spot specifically because you probably haven't gone over it yet, but it's been a while since we went over it. But you know inhibiting hormone, you know releasing hormone. That's what I mean by just sorting the hormones. Okay? If you've got an inhibiting and releasing, it should make sense to you that it should be a G alpha S, G alpha I combination. Okay? So even if you don't know this, and I really don't expect you, I don't expect you to be able to just jump out and say, this hormone goes with this receptor right away. But I think in a case like this, you should be able to reason it through and come up with an answer on your own. All right. Back to items. Let's do another one. Okay, this one's the first two were pretty easy. This one's a little harder. And this is the kind of question that kind of separates the A's from the B's on the test. Did somebody just say darn you to me? Or okay, I just heard darn you out of the audience. I've actually heard worse. Actually, a lot worse. Okay, you guys seem pretty confident. Have you answered it already? Or oh, okay, good. Okay, so how many think you got that one right? Yeah, this is a much harder question. So, okay, so let's take a minute and walk through this one. All right. So this is good. So a lot of people jump on this one, the bacterial infection. So we haven't talked about bacterial infection at all. But if you see something like that, connect it to what we have talked about. So if you hear bacterial infection, you should be thinking immune system, right? And if you think it through, glucocorticoids actually increase your chances of getting an infection. That's one of their problems. They suppress the immune system. So people who are taking high-dose glucocorticoids, for whatever reason, often have to be extremely careful about picking up some sort of illness. Okay? Hypoglycemia is just getting the difference between hyper and hypo mixed up. Be careful, right? Remember, Cushing's disease is high cortisol or high glucocorticoids. It's a diabetes-like condition. Diabetes is hyperglycemia. Okay, so those are the dots you connect there. Elevated response to epinephrine is the response is a property of the target tissues, okay, not epinephrine itself. Thyroid hormone was just because I couldn't figure out what to put in. A lot of people, when they're lost, just go for the one that they haven't any idea about. That's typically a bad strategy. If you say, gee, I'm not sure which one of these four that are connected to the question might be it, so I'll just go to the one I have no clue about. And so, yeah, the reduced response to ACTH. That's good, though. 60% is pretty good. And this, again, when you go through the practice test, these are the questions that give people the most trouble, and the people who get A's handle this kind of question. The 2 or 3 of them will get A's. God, that always gets a big response. I say the 2 or 3 people who get A's, and I get a lot of more than 2 or 3 people get A's. But more than 2 or 3 people get A's, but in 5 years on exam 1, I think I've had 10 people get perfect scores. That's the bad news. The good news is half of them were last year, so maybe you guys are getting smarter or I'm getting soft. Although nobody, if you include exam 1 in the final, in 5 years, I've never had a person get a perfect score. Any of you gunners out there looking for bragging rights. Okay, so now we'll go with endocrine regulation of fluid and ions. And a lot of this will touch on some of the things you went over in when you did renal. And I won't talk about function very much. This is mostly going to be a talk about regulation and how these different hormone systems interact with one another. We'll center it around, although there are other things involved, I'm centering around 3 hormones: antidiuretic hormone, angiotensin, and aldosterone, and how the 3 of these integrate. So fluid and electrolyte levels in the bloodstream are carefully regulated. There are electrolytes in the extracellular fluid, sodium, potassium at much lower concentrations, and the osmolarity has to be maintained between 280 and 296 milliosmolar. Okay. And again, this regulation is more complex than just the endocrine factors. Okay. To regulate the system, then, the kidney is, of course, responsible for maintaining water and ion levels. The vasculature determines blood pressure or blood volume. They're actually 2 sides of the same coin. Okay. And the hormones are antidiuretic hormone, ADH, angiotensin II, and aldosterone. And again, one of those funny little ways medical students have to remember things. Think about the A team. Old school A team. This new stuff was nonsense. Okay. Antidiuretic hormone. Okay. Abbreviated ADH, it's more often known as vasopressin. Okay. That's what the renal people call it. But I think antidiuretic hormone is easier for us to remember, so I'm going to go with that name. It is secreted from the posterior pituitary. So this is our first posterior pituitary hormone. Okay. Meaning that the hormone is synthesized in cell bodies in the hypothalamus. Axons extend down the length of the posterior pituitary, where ADH is released directly into the blood supply. Its regulation is through neural inputs in the hypothalamus. ADH is a 9 amino acid peptide. its receptor is is coupled to G alpha Q if it's in the vasculature, or G alpha S if it's in the kidney. Its major effect is water conservation. That's the way to think about it. Although, like everything renal, it's sort of complicated. Okay, so antidiuretic hormone is secreted in response to sensory input. So a decrease in water concentration, a decrease in pressure or volume in the vasculature, thirst, all stimulate release of ADH. Also, the hormone angiotensin II stimulates release of ADH into the bloodstream. By the same token, an increase in water concentration, increases in pressure or volume, inhibit ADH release. Okay. The key input is the water concentration, or the osmolarity. There's actually an osmolarity sensor in the brain. So ADH in the kidney works to conserve water. It binds to a G-alpha S coupled receptor, leading to active protein kinase A. Protein kinase A then stimulates a transcriptional program that synthesizes a channel called aquaporin, which is a water channel. Protein kinase A also stimulates the transport of aquaporin to the surface of the lumen, where it allows transport of water into the cells and eventually into the bloodstream to increase the water concentration, or decrease the ion concentration in the bloodstream. In the vasculature, antidiuretic hormone increases blood pressure and blood volume. Okay. So there, the receptor is coupled to G-alpha Q. Okay. G-alpha Q activates phospholipase C beta, giving an inositol triphosphate, which binds to a calcium release channel, releasing calcium into the cytoplasm. That calcium then interacts with the muscle directly to cause contraction of the vasculature, increasing blood pressure. Okay. So it increases blood pressure by decreasing the vascular volume through contraction. Okay. And this is mostly a response to severe bleeding. It's kind of a stress thing. Angiotensin is much more important in this. So next we have the renin-angiotensin system. Now your heart should be beating faster. Angiotensin II is an 8 amino acid peptide hormone synthesized from a precursor called angiotensinogen, which is a 485 amino acid protein, which is made constitutively in the liver. So constitutively means all the time. Okay. It's then converted by proteases in the bloodstream to angiotensin I, which is 10 amino acids, and then activated by a second protease to the 8 amino acid angiotensin II. It has a short half-life, again about a minute, 2 minutes, where a third protease degrades it to angiotensin III, inactivates it. And some people think angiotensin III has some effects, but that's controversial. We won't go over those. So this is the layout of the renin-angiotensin system. Now the proteases are renin, which is secreted from the kidney under the stimulation by a decrease in blood pressure or blood volume. And it's inhibited by high levels of angiotensin II in kind of a feedback here. This is a feedback that doesn't involve the central nervous system. The key enzyme to convert angiotensin I, so renin goes from angiotensinogen to angiotensin I. Angiotensin I is converted to the active angiotensin II by a protease called ACE, or angiotensin converting enzyme. Okay. And it's a major drug target for controlling blood pressure by inhibiting the angiotensin II response, because it's not part of any regulatory system. And the problem with most endocrine regulation using endocrine analogs like glucocorticoids as drugs is you have all these problems with feedback, with the whole feedback response. And so there's all these deleterious effects. The beauty of angiotensin, of ACE inhibitors, is that ACE isn't involved in any of that feedback. So you don't end up messing up 50 other things in the process of trying to regulate one hormone. Okay, so angiotensin II Binds primarily to G-alpha Q and G-alpha I coupled receptors. It constricts blood vessels, which is similar to ADH, but it's a much more important signal for blood vessel constriction or contraction. In the kidney, it inhibits renin release and stimulates the reabsorption of sodium. It also stimulates, as I mentioned before, the secretion of ADH, and also stimulates this thirst response. And finally, it's involved in increasing the synthesis of our third hormone, aldosterone, which is made in the adrenal cortex. Aldosterone is a steroid. It is the primary mineral corticoid. Okay, so this is a similar situation to the glucocorticoids. In some animals, there are other mineral corticoids that are active, and there are chemicals you can find that act like mineral corticoids, but the only one worth remembering or that's really active in humans is aldosterone. And this terminology is sort of a leftover from when people were trying to characterize how this activity worked. Okay. It's made in the adrenal gland, in the zona glomerulosa, which is kind of easy to remember because it sounds like it's kidney related, right? And it's up here close to the venous circulation. It's synthesized from corticosterone by an enzyme unique to the zona glomerulosa called aldosterone synthase, which is easy to remember. But you don't have to remember it, it'll just come up. And aldosterone is a stress hormone, like the other hormones released from the adrenal gland. So aldosterone's targets and functions. It doesn't have that many well-recognized targets. Okay. Its major target is the kidney. It also has minor targets, including the colon, sweat glands, and salivary glands. Its functions then are to promote potassium excretion and sodium reabsorption. But its real physiological role, there's not enough aldosterone receptors in the kidney to have much of an effect on sodium. Okay? So its primary physiological role is potassium homeostasis. Okay? And so that people who have trouble making aldosterone, their primary issue is with potassium homeostasis, not sodium. So, and that's how you surmise that's physiologically what is key in. Okay? So hypoaldosteronism can result in hyperkalemia. Kalemia is like potassium if you see the K. So high potassium levels, which can be quickly fatal because they alter the way the heart works. Aldosterone is similar to cortisol. So you see these two steroid hormones. If you take the three-dimensional structure, see they've just got this little difference here. If you take the three-dimensional structure, they don't look that much different. I mean, I tried to sort of twist these around so they look pretty similar. They're also similar in the way they get synthesized, except for that one enzyme at the end. Their signaling mechanisms are similar through transcription. There are some important differences. Aldosterone is the one steroid that we're going to go through that has an important non-transcriptional signaling mechanism. So it does both. It's also pretty soluble. It's the most soluble of the steroids found in the bloodstream. So it doesn't really need, most of it is free. Okay? And therefore, for a steroid, it has a pretty short half-life, about an hour. So the cortisone and the aldosterone receptors are similar. They're part of the same gene family, and they're very, very close to one another in terms of sequence. So that the binding affinity of cortisol for the mineralocorticoid receptor, so here's aldosterone, here's cortisol. The binding affinity for cortisol to its receptor is about the same as cortisol to the mineralocorticoid receptor. Okay, so how do we tell them apart? Okay. This is a real problem because the average circulating concentration of cortisol is 2,000 times that of aldosterone. Right? So why have aldosterone at all? Why doesn't cortisol just substitute for aldosterone? Okay. How could aldosterone ever have an effect? Cortisol binds the receptor just as well, and there's much, much more of it. And the reason is that true target tissues for aldosterone inactivate cortisol. All right, so here's cortisol. And those tissues have an enzyme called 11-beta-hydroxysteroid dehydrogenase 2. Again, I won't try to catch you on the difference between 1 and 2. And that converts cortisol to inactive cortisone. And this reaction is irreversible. Okay. The cortisone is soluble, so it just ends up in the bloodstream, is glucurinated or sulfated and excreted from the kidney. Okay. Aldosterone does not substitute for cortisol because the circulating concentration is much lower and because aldosterone has little affinity for the glucocorticoid receptor. So this is only one way. All right, and that gives you an idea how complex the interactions can be with these similar receptors. And there's other examples of this kind of thing, but I think this is the easiest one to deal with and probably the clearest and most important one. So there are two signaling mechanisms for aldosterone, a slow one and a fast one. Okay, the slow one, here's aldosterone. It binds to the mineralocorticoid receptor, activates a transcriptional program, which stimulates the synthesis, this is in the kidney, of pumps, channels, transporters, oxidative phosphorylation to make ATP, all the things needed to require to reabsorb, to increase potassium excretion. I'm getting my reabsorption, my excretion mixed up. Okay. So it essentially increases the activity of potassium excretion in the cells in the kidney in which it targets. There's also a protein in these cells called the serum glucocorticoid kinase. Don't ask too many questions about it. In fact, the name has so little to do with what it does. Everybody just calls it SGK. Okay, so the SGK binds aldosterone, it's a kinase, and activates it in the cytoplasm. And then that activated kinase causes the transport of sodium and potassium channels to the cell membrane to allow the excretion of potassium and the reabsorption of sodium. And this was discovered based on timing, okay, that this transport of these channels to the surface was way too fast to be accounted for through a transcriptional mechanism. That's how people started looking for it. Okay, so the result is to increase potassium excretion and sodium reabsorption. So that the synthesis of aldosterone then is integrated through a lot of factors. We went through all these hormones that are involved in blood pressure regulation and volume and potassium levels. Okay, so hyperkalemia is the most important regulator. And hyperkalemia changes the voltage gradient across the membrane, activating a voltage-gated calcium channel, which allows calcium to come into the cells. Antidiuretic hormone binds to G-alpha-Q coupled receptors on the zona glomerulosa cell surface, activating G-alpha-Q, as does angiotensin II. Those serve then to release calcium from intercellular stores and activate protein kinase C. There's also ACTH receptors, which activate protein kinase A. Now, all the effects you're going to see are some combination of these things. Don't worry about which is which. In fact, it's not that clear which is which. Okay? But the combined effect of calcium, PKC, and pKa results in a general upregulation of steroidogenesis in these cells. So there's direct activation of STAR and aldosterone synthase, increase in transcription of the side chain cleavage enzyme, steroid acute regulatory protein, and the aldosterone synthase to give higher amounts of aldosterone. So the endocrine control is mostly self-regulating. There's a bit of nervous system involvement here. But this is a system that's mostly just on autopilot and regulates itself. All right? So we have the hypothalamus and pituitary, the adrenal gland, kidney, and angiotensinogen out in the bloodstream. So a decrease in water volume, an increase in potassium concentration, or decrease in blood pressure and blood volume, or decrease in sodium concentration, which are all, of course, related. stimulates secretion of renin to allow conversion of angiotensinogen to angiotensin II, which is then quickly converted into angiotensin I, which is then converted to angiotensin II. Angiotensin II plus the water and the pressure volume stimulate the release of antidiuretic hormone. Angiotensin II stimulates thirst, as does the decrease in water volume. That also stimulates antidiuretic hormone. Thirst is also a stress. It directly stimulates the release of CRH. It's not that important effect, though. And ADH and CRH stimulate the release of ACTH. Angiotensin II, then, as well as the hyperkalemia, ADH, and ACTH stimulate synthesis of aldosterone. Aldosterone then decreases potassium concentration, along with angiotensin II increases sodium concentration, increases blood pressure and volume. Antidiuretic hormone increases water volume. It also stimulates the desire to drink. It's sort of a behavioral input. Actually has a lot of complicated behavioral input, but this is the important one. Okay. The feedback from ACTH to CRH reduces that response. Angiotensin II inhibits renin, which inhibits further synthesis of angiotensin II, which reduces the stimulatory effects to ACTH and CRH and thirst. The antidiuretic hormone then, you start to lose the effects of aldosterone synthesis, ACTH goes down, and you end up with a happy homeostasis reestablished. All right? So good. I will see everybody Thursday. Yes. Let me collect my stuff.